Good day everyone, this is Rijal Aligado and today we are going to talk about meiosis. So first, what is meiosis? Meiosis is the process whereby chromosomes are copied, paired up, and separated to create eggs or sperm cell. So before meiosis begins, some important changes take place within the parent cells. First, each chromosome creates a copy of itself. These duplicated chromosomes are known as sister chromatids. They are fused together, and the point where they are joined is known as the centromere. Fused sister chromatids roughly resemble the shape of the letter X. Meiosis occurs over the course of 2 rounds of nuclear divisions. It is called Meiosis I and Meiosis II, according to Nature Education Citable. Furthermore, Meiosis I and II are each divided into 4 major stages: prophase, metaphase, anaphase, and telophase. Meiosis I is responsible for creating genetically unique chromosomes. Sister chromatids pair up with their homologs and exchange genetic material with one another. At the end of this division, one parent cell produces 2 daughter cells, each carrying one set of sister chromatids. On the other hand, Meiosis II closely resembles mitosis. The 2 daughter cells move into this phase without any further chromosome duplication. The sister chromatids are pulled apart during this division. A total of 4 haploid daughter cells are produced during the course of Meiosis II. According to Molecular Biology of the Cell, Garland Science 2002, there are 4 stages in Meiosis I: prophase I, metaphase I, anaphase I, and lastly telophase I. First, let's discuss the prophase I. At this stage, chromosomes become compact, dense structures, and are easily visible under the microscope. The homologous chromosomes pair together. 2 sets of sister chromatids resemble 2 X's lined up next to each other. Changes bits of DNA with the other and recombines, thus creating genetic variation. This process is known as crossing over or recombination. By the end of prophase I, the nuclear membrane breaks down. Next is metaphase I. The meiotic spindle, a network of protein filaments, emerges from 2 structures called the centrioles, positioned at either end of the cell. The meiotic spindle latches onto the fused sister chromatids. By the end of metaphase I, all the fused sister chromatids are tethered at their centromeres and line up in the middle of the cell. The 2 homologs still look like 2 X sitting close together. In anaphase I, the spindle fibers start to contract, pulling the fused sister chromatids with them. Each X-shaped complex moves away from the other toward opposite ends of the cell. However, lastly, in telophase I, the fused sister chromatids reach either end of the cell and the cell body splits into 2. Now, Meiosis I results in 2 daughter cells, each of which contains a set of fused sister chromatids. The genetic makeup of each daughter cell is distinct because of the DNA exchange between homologs during the crossing over process. However, in Meiosis II, there are also 4 stages. It starts from prophase II. The nuclear membrane disintegrates and meiotic spindles begin to form once again. It is followed by metaphase II. It is where the meiotic spindles latch onto the centromere of the sister chromatids, and they all line up at the center of the cell. Next is anaphase II. The spindle fibers start to contract and pull the sister chromatids apart. Each individual chromosome now begins to move at the either end of the cell. Lastly, is the telophase 2. The chromosomes reach opposite ends of the cell. The nuclear membrane forms again, and the cell body splits into two. Meiosis II results in 4 haploid daughter cells, each with the same number of chromosomes. Now, why is meiosis important? Proper chromosomal segregation, or the separation of sister chromatids during meiosis I and II, is essential for generating healthy sperm and egg cells, and by extension, healthy embryos. If chromosomes fail to segregate completely, it's called non-disjunction and can result in the formation of gametes that have missing or extra chromosomes. Moreover, the most significant impact of meiosis is that it generates genetic diversity, and that's a major advantage for species survival. That's all. Thank you for watching, and again, don't forget to subscribe and like my channel.